Uh, I am speaking on behalf of Beatriz Mintz. She was born in 1921 and died this year, just 3 weeks before her 101st birthday. Her family emigrated to the United States from what is now Ukraine. And like many accomplished women of her generation, she chose career over family. In the early 50s, when she started her career, there were very few women in science. She received her PhD from the University of Iowa, was a professor at the University of Chicago in a teaching-focused position, but then switched to and moved to Fox Chase Cancer Research Center in the 60s because there she could devote 100% time to research. Her personality has been described as efficient, organized, self-sufficient. She built all her own equipment, and she was known to be demanding but a kind mentor. She also had a softer side and a sense of humor. She wrote poetry featuring mice, and she built a huge snow mouse in the winter. She had a big impact on our understanding of development and how complex tissues develop from a single cell. She, at the same time as Ralph Brinster, made the first chimeric mice. These were made by combining early embryo cells of two genetically different mouse strains. And this was used to establish the clonal origin or single cell origin of lineages as diverse as pigment, tissues, somite, muscle, vertebrae, the skull, and hematopoietic lineages of the blood. She also demonstrated that just a handful of embryonic stem cells generate the remarkable complexity of the fully developed animal. She generated the first transgenic mice by injection of DNA into blastocysts and later into the pronucleus. She addressed major questions regarding cancer and how the proliferation of cancer cells is affected by their microenvironment. She placed tumorigenic teratoma cells into a normal mouse blastocyst and showed that the tumor cells became normalized by the embryonic environment and were able to generate normal tumor-free mice. This was remarkable at the time, and it argued that cancer is not only caused by irreversible genetic changes, but also by reversible epigenetic alterations. And that's a branch of cancer biology that is actively studied today. Using these mouse models, she also studied melanomas, and her transgenic mouse model showed that melanoma tumor cells metastasized the skin and the eye and showed the complexities of this interesting tumor and how it develops. She received a number of awards, among them election to the National Academy of Sciences, election to the American Academy of Arts and Sciences, the first prize in developmental biology from the March of Dimes, together with Ralph Brinster, the National Medal of Honor for Basic Research from the American Cancer Society, and a Lifetime Achievement Award from the American Association for Cancer Research. She was also an elected member of the Pontifical Academy of Sciences. And interestingly, in the obituary of B. Mintz that was published in the Cancer Letter this year, written by Jonathan Chernoff, the director of the Fox Chase Cancer Center, he said, quote, "More than any of all these honors, Beatriz Mintz was proudest of having been elected to the Pontifical Academy of Sciences, where she advised Pope John Paul II about embryonic stem cells." I knew and admired B. Mintz. She was a force of nature and a pioneer. As written in PNAS in the obituary published recently, she will be remembered as a giant in mammalian embryogenesis and developmental genetics who laid the foundation for our present understanding of mammalian development.